sort of transmembrane protein. Like, for example, in this illustration here, you can see it's a G protein coupled receptor. And so this particular receptor protein will pick up some sort of inducer, some sort of chemical message from the outside. And what will happen is when it receives that message, it'll change shape. And the reason that it's called a G protein coupled receptor is that it associates, when it changes its shape, it associates with the G protein, and that is a trimeric protein, and that changes shape. And what happens is you get a cascade of events. Once the G protein is activated, you get a series of other proteins that become activated in what's known as the signal transduction pathway, and this occurs inside the cytoplasm. So it involves secondary messengers, it involves calcium, it involves cyclic AMP, it involves molecules that, while in the cytoplasm, can amplify the message, and then the cell can respond most efficiently. And so that's what this video is going to attempt to discuss. That is, it's going to try to emphasize signal transduction pathways. And so, as I was saying, it's a cascade of molecular interactions in the inside of the cell, but it starts from a signal from the outside, and that's picked up by a receptor. And then the signal transduction pathway, which involves a few steps, will amplify and regulate, as we shall see, the cellular response. And so let me give you just a simplistic, believe it or not, a simplistic example of this. And so when the receptor picks up a message, maybe the particular message is a molecule changes shape. It's often an enzyme, and that particular enzyme becomes active. And so if it's a kinase enzyme, and I'll go over this in a moment, if it's a kinase enzyme, it's going to transfer a phosphate to another protein in a process called phosphorylation. And that usually changes the shape of that protein, which then changes the shape of the next one, and the next one, and the next one, and the next one. And these things increase in concentration, therefore amplifying the signal. So just a little bit of message goes a long way inside. And you're like, well, then what's going to happen? Oftentimes, what will occur is that a molecule will go inside the nucleus of the cell. And basically, this gets detailed, but it's pretty cool. It's not the real topic of this video. But these molecules can interact with transcription factors. In other words, by phosphorylating a transcription factor, that engages a particular promoter region of a gene, which is where RNA polymerase binds. And therefore, a gene is then turned on, or maybe it removes a molecule that's inhibiting a regulatory protein. So it can either turn on or turn off a response. And so if the gene is turned on, let's just say, then RNA polymerase will transcribe that gene into messenger RNA. So a lot of RNA is then produced. It goes into the cytoplasm. And then that cell's ribosomes will make a whole slew of enzymes that weren't being made before. And you're like, well, what's that going to do? Well, that's going to respond. The cell's going to respond to that. And so there's some pathways that are going to be turned on as a result of that. Perhaps the cell initiates division as a consequence of receiving this message. And so the transduction pathway is what we want to focus on today. And it's usually multiple steps. And when it's multiple steps, that affords an amplification. And it also affords regulation and better coordination internally. So let's take a look at that. So when you think of molecules relaying messages from a receptor, these are mostly proteins that we're talking about in the pathway. Protein is activated, then that can become a molecule that activates other molecules. And then when those molecules become active, those can activate other molecules. And so it not only is it sort of a cascade, but it also is an amplification. So I want to emphasize this point because not only is it important, I find it particularly cool and useful. And it explains a lot about what transduction really is trying to accomplish, which is amplification. So let's say, for example, we're talking about the fight or flight response. And so somebody's chasing an animal, and epinephrine is released from the adrenal glands, and it's targeting a muscle cell or a liver cell. And the goal is, the response is to get the polysaccharide glycogen to release sugar in the form of glucose to raise blood sugar levels so that the animal can respond and run away. And so how that is accomplished is that the receptor protein picks up the epinephrine, and what that will do is change the shape of the receptor protein and basically activate the inactive G protein, because this particular receptor is a G protein coupled, so coupled meaning associated with receptor proteins. And so when that becomes activated, many of the G proteins become activated, and as you can see right here, one molecule, this is what I want to emphasize, that epinephrine can produce 10 to the second power of activated G protein, so that's a big amplification. Then this activated G protein will activate and adenylate cyclase. And this is an enzyme that's also membrane bound. And don't get caught up in the detail, but once that becomes activated, this molecule will then be able to convert adenosine triphosphate, ATP, into a copious amount of cyclic AMP. So it's the removal of the pyrophosphate, yada yada. But the cyclic AMP acts as a secondary messenger, And so what is that doing? Well, cyclic AMP now has 10 to the 4 power, so the concentration of cyclic AMP is rather large in the cell compared to 1 epinephrine. So check that out. So then cyclic AMP is capable of activating these protein kinases, which are plentiful enough in the cell, but they're inactive. And then when they become active, the kinase is an enzyme that is capable of transferring a phosphate. So kinase A gets activated, and then that activates another enzyme called phosphorylase kinase. So phosphorylase kinase becomes activated, and look, the numbers are increasing even more, 10 to the 5th. Okay? So 10 to the 5th. And then what is this doing? The inactive, that phosphorylase kinase activates, this is the crucial enzyme in what is going to be involved in the response. The enzyme that we really want going on in the cell is glycogen, which is a polysaccharide phosphorylase. So what does that enzyme do? That's capable of hydrolyzing or cleaving glucose off of glycogen. And so that enzyme becomes active, and that's the point. And so look at this. So many of those enzymes. And so therefore glycogen is just going to get mowed up, it's going to get chopped up. And then the consequence is look at all of that glucose. So the cell is just spewing with glucose. It's just pouring out 10 to the 8 molecules of glucose 1-phosphate. And so that ultimately will diffuse out of a cell and raise blood sugar level. And so pretty cool. So you get a ton of glucose being released from just one little message. So signal transduction, response, but ultimately the signal was amplified. So each step of signal transduction is sort of the transduced part of it. Usually there are different exceptions. It usually involves some sort of shape change. And so just to emphasize this, if I have sort of a molecule like this, and it's a protein, and it's what we call inactive, usually what this involves is some kind of, when a phosphate is stuck to it, this is the negatively charged functional group, it usually results in activation. And so what's happening here is that the protein is literally, I'll try to emphasize this here, it's literally becoming something useful, and so it's now capable of doing something. And what's really cool about this is sometimes we want enzymes to be active, sometimes we don't want it. So it goes both ways. Like it's not always appropriate to activate, sometimes it's appropriate to inactivate. And so along those lines, what we're talking about is 3 things. We're talking about reception, we're talking about how proteins become activated,